Okay, all right. Thank you everybody for coming. My name is Swain Chen. I'm from the Genome Institute of Singapore, GIS. I'd like to tell you today about the research that's being done in my lab as well as the rest of the institute, where mostly we're studying genomics and how that can help us, in particular with understanding infections today, and may help us improve our healthcare and our lifestyles in the future. So when I talk about infections today, maybe we can start off with an outbreak that actually happened in Singapore pretty recently. In 2015, there was an outbreak of infectious disease in Singapore, and as is appropriate for the 2010s, the news of this was actually broken in WhatsApp on social media. And so I'm going to go through this because I think it does a great job of highlighting some of the really unique aspects of this infection, of this outbreak in 2015. So this WhatsApp message says that he's sharing this because a boss is warded in the hospital from a painful right arm. His boss ate raw fish at a hawker center and then became sick and now wants to share this with as many people as possible. For the past few weeks, hospitals have been seeing a surge of young and old patients coming in with fevers and painful joints, and these have been caused by a particular strain of bacteria called Group B streptococcus. Okay, so I want to go through each of these three in order, but we'll start with the bottom. Group B streptococcus. What is Group B streptococcus that was causing this outbreak? We know about GBS, which is the abbreviation for Group B streptococcus. We know about this in medical teaching. It does cause infections of humans. It causes infections mostly of newborn babies, where it oftentimes will infect the linings of the brain, an infection called meningitis. And it also infects elderly patients or people with diabetes, people who have compromised immune systems. These are the things we know about. These are the things we look for when we see a GBS infection. What we don't look for is a connection to eating food. But in that year in Singapore, in 2015, the food-associated infections were much more than all the other infections that we expect from GBS combined. Okay? So this was totally new because GBS, as I mentioned, was never associated with food before. This is something we don't look for, something we don't associate with GBS infections. So that's one of the issues that we had to solve when we're trying to study this Group B strep outbreak. The second thing I mentioned to you was that there was a surge of young and old men. So what does that look like? I'm showing you here a graph of GBS infections over 2014 and 2015. These are weekly case numbers, weekly infection numbers. And this gray area here sort of looks like a background rate of infection. Nothing much is going on. In 2015, what happened was we had a surge of cases, and it looked like this. And that extra part, the extra height of these bars was all due to the fish-associated infections. Okay, so this is what a surge of infections looks like. And of course, when you see these numbers go up so dramatically from 3 or 4 over here up to 30 or 40, of course you see that something is going on. And this is what the public health agencies and the food agencies were looking at and why they knew they had to go figure out what was happening. When they figured that out, that it was associated with fish, then they put some controls in place and the infections came down. Okay, so when there's a surge of infections, you know something's happening. You know an outbreak is going on, and then a whole lot of processes get activated for us to investigate this outbreak. And this is what happened here, but if you look, to detect an outbreak through a surge detection, we have all these patients that are already getting sick before we know what's going on, before we can figure it out. And what we'd like to do, and one of the things that in my lab we're trying to work on, are methods so that we can do the detection over here. And what I want to tell you today is that one of the technologies that can help us do this is called genomics. So what is genomics? Now I know not everyone in here may be a biologist, so to make sure we're on the same page, maybe you can remember back to biology class in grade school or high school, and hopefully you remember that all organisms, including humans, are made up of cells. All organisms, including humans, are made up of cells. Inside of cells, every cell has a nucleus. And inside the nucleus, we have what we call chromosomes, and those chromosomes then, in turn, are made of DNA. Now, if you take the total set of DNA, the total set of chromosomes inside of a nucleus, that total set of DNA is called the genome of the organism. And of course, what we do is genomics, which is the study of these genomes. Now, you may have heard of genomes or DNA being referred to as the book of life, or you may have heard of DNA being used for paternity tests. And the reason we can do that is because humans have human DNA, and that makes us humans. Other animals, say like lions, have lion DNA, that makes them lions, and that DNA is different. And if you look around you, there's other people in the room. But you're different from all the other people in this room. And that's partially because your DNA is also different from the other people in this room. So we can use the DNA, we can use your genome to identify individuals and be very accurate in terms of who is who or differentiating between different people. And we can do that not only for humans, but we can also do that for other organisms like bacteria, like the group B strep that caused this outbreak in 2015. So one note, just like humans are all different from each other, group B streptococcus, we refer to this as a bacteria, but they also have different individuals. They have different DNA as well, and there are different group B streps that we can differentiate between also. And that's how we actually use genomics to help understand these two issues that I just noted for you, the association with fish as well as the surge of cases. So how does genomics actually help in this case? So the big question was we didn't know if this was real. We'd never seen GBS infections associated with food before. So is it really that this human got sick from eating this fish? Well, the way we solve that, or one of the things we can do with genomics, is the patient is sick, and so we can take a blood sample and isolate the bacteria that's in that blood. We can then go to the Hawker Center and take a sample of the food at the same time or the same place that that person was eating, and we can also isolate bacteria from that food sample. Now that we have these bacteria, we can sequence their genomes or look at the DNA that's inside of them, and then just like we do for paternity testing or something, we can tell whether those genomes are the same. We essentially get a genomic fingerprint for each of the bacteria from the human as well as from the fish. And if those two match, then it's the same GBS. It's the same individual, right? And then what we can do is combine with some other epidemiology data or medical data, we can make the connection and say, yes, indeed, it was the case that this person got sick by eating the contaminated fish. So that's how we can help solve these new outbreaks where we don't know that this is how this disease occurs from previous experiments. But what about the surge of cases? Okay, how do we deal with that? How is genomics going to help us prevent these infections? Well, if you're in a hospital and if we're doing this, we isolate the bacteria from the blood of a patient and we get that genomic fingerprint from that bacteria. And say a few hours later, another patient comes in and we do the same process. They have the same disease, and actually it turns out they have the same genomic fingerprint for their bacteria. And then if a few hours later, a third patient comes in and again, you see the same genomic fingerprint. Now you can kind of piece together, well, this looks like the same thing is happening, right? It looks like a mini surge of infections. We've connected the same culprit or the same criminal at 3 different crime scenes. And so this is something that we'll have to now start to activate the process of going to examine what's happening. And so that's how genomics can actually help us to detect the outbreak at this stage right here, just with a few infections. Because what I didn't tell you is that all the red infections are caused by the exact same individual, the exact same group B strep, whereas the gray ones, those are all different ones, right? Those are all different, they're not related at all to each other. Okay, so that's how genomics can help us to save infections. It can help us to confirm the source that's unknown for an outbreak, as well as prevent this surge of cases. And in this case, we could have saved, had we, in retrospect, looking back, we know that genomics could have helped us save about 300 infections of GBS. Okay, so that's fine. In retrospect, we can do that. But outbreaks occur at unexpected times. And I already told you that this outbreak occurred with an unexpected cause, with association with food. So how do we prepare for the unexpected? Right? If we know where to look, of course, that's okay. Well, maybe what we can do is take a page from another arena where we try to anticipate or we try to find unexpected events, say crime or accidents. And the way we try to do that, the way we try to detect crime or accidents is we constantly monitor. We have cameras all over the place that are continuously recording. And that's how we find unexpected events. So you'll see cameras like this. I'm sure you guys have seen these around town. You see these in taxi cabs and other cars. These are constantly recording, looking for unexpected events. And I think if you watch the news a little bit, you'll start to see more and more. You'll have user cell phone videos that come because the events that happen in the news, they're all unexpected. And who's got recording devices there? The general population, because we're all carrying these around in our pockets. Okay? So we monitor continuously and ubiquitously. And the question, though, is can we do that with genomics? Could we actually just be sequencing everything all the time? And again, before I get to that, maybe we should continue with this analogy and ask the question, how is it that we're able to use digital devices to monitor all the time and everywhere? And a lot of that has to do with the fact that computers, from the 1960s to the 2000s, over 40 years, they've gotten smaller and faster. These things used to take up half a room. Only the biggest companies or governments could own these computers. But now, essentially everybody in this country has at least one or maybe sometimes 2 or 3 of these that they're carrying around. That process took 40 years for digital devices. In genomics, a similar process is happening. This is a genome sequencing machine that was available in 2000. Again, it took up half a room. And this is still in our institute today. But 4 years later, this device became available on the market. And it's something that you can hold in your hand. Okay? And so the size reduction that happened in computing devices, that size reduction that took 40 years has happened in 4 years in genomics. And I think the same things that this is driving, where now instead of just companies and governments, now the average person can have these, this is going to drive the same process. And actually, it's happening much faster in genomics than it happened with computers. So if it becomes a daily device, if it becomes something that is just another part of your cell phone, what does that mean? How is our life going to change from that? Well, I told you what genomics can do in terms of helping us with infections, right, with this GBS outbreak that happened in 2015. There are other healthcare applications that genomics can help us with as well. It's not just infections. One of the more famous examples is Angelina Jolie, who she had a DNA test which told her that her risk for getting breast cancer was much higher than the general population. So then she was able to make a decision to have a double mastectomy, to have a surgical procedure to reduce her chance of getting breast cancer. And that's something that then, can sort of save a lot of grief and save a lot of lives that way. And while this technically is not a genomics test that she did, it was just a DNA test, all the other DNA tests that we're trying to discover right now for all the other cancers to do the same kind of predictive risk assessment, all of those today are being discovered using genomics. So that's how it can help with healthcare. I mentioned as well how you may have heard of DNA being used for paternity testing, or how we use DNA to identify different individuals, which makes it a perfect technology for identifying your relatives or for doing genealogy. And so this is now starting to be not really a healthcare application, and it goes even further than that. So just as an example, there's a company called Vinome. Okay? Vinome takes your DNA. What they offer is to take your DNA and they'll sequence it. And based on your DNA sequence, they can predict what flavors you best appreciate or what flavors you're going to like. And from there, they want to take that flavor profile and predict, this is the wine that you should try because you will best appreciate the flavor profile in here, right? Okay, so I see some smiles here. It goes on. There are companies that want to take your genomic information and tell you what you should eat to build muscle or to lose weight or to stay healthy. There's companies that go even further. Soccer Genomics wants to use your genomic information and build you a customized training program so you can be better specifically at playing soccer. Okay, and then it goes on even further after that. There's another company, and this is pure entertainment, right? It's just give us your DNA, give us your sequence, and what we'll do is we'll take that sequence and convert it into a customized color or customized pattern that is uniquely you. And we can make you a t-shirt or a pair of socks that then you can demonstrate your genome sequence. Okay, so I see the reaction that you guys are having, which is exactly correct. The point I want to make right now, though, is that there are companies out there that are thinking about how genomics can be used, not just for healthcare, right? Not just for research purposes, but for your daily lives, as a lifestyle technology, as a consumer technology, okay? And while you guys may have some ideas as to how good these companies might be or how good the technology is at this point, I think that's actually the point. This is exactly how I think about it as well. Because we know from the internet, from internet companies, it's always been a process of trying out new ideas and then figuring out what works. Okay? There will definitely be fits and starts, and we're probably in that fits and starts area. So you guys may have heard of a company called Webvan, which was opened in 1996. In the US, Webvan, their idea was to do online grocery shopping. You log onto the website, you log into your account, you click on a few things, and then those groceries show up fresh at your door. That company ended up going bankrupt and closing within a few years. Okay, but I think a lot of you in Singapore are probably thinking, but that exists now. We have Redmart. And in the US, there's another company, Amazon Fresh. Their idea is you log onto the internet, you pick your groceries, and then they show up fresh at your door. It's the same idea and concept. But what's happened is some of the executives from Webvan, after that business went bankrupt, some of them have now tried again, and they're helping to run the business in Amazon Fresh, and that's doing much better. And I think those of you who are using Redmart know that that's not a bankrupt company right now. Okay, so we do have fits and starts, but I think eventually we kind of figure this out. Just because Webvan failed does not mean that the promise of the internet or the promise of these kinds of, the way that the internet can pervade your daily life, that doesn't mean that that promise was false. Okay, it just meant that Webvan didn't succeed. And what I think happens is there's a lot of maturation, both in terms of the technology gets better, but our understanding of that technology and how to apply it to different industries or different domains, that also gets better, and that gets more mature. And I think if you think about companies on the internet today that are really succeeding, they're not failing at all, these guys are very mature in how they use technology and the internet to deliver their products. The last thing I want to highlight here is that the consumer is an extremely important part of this. Another big difference between the 1990s and today is that consumers also are much more familiar with the technology, with internet technology, right? They're much more familiar with how that can be useful in their daily lives. And that also helps these companies, and that whole ecosystem helps to put the entire thing together where this technology then ends up transforming our lives. In genomics, I think that's what we're looking forward to. In genomics, we're still in the early years. We're in the early stages, but we're going off into this future. And I think the path forward, right, is we need this ecosystem here so that we can gain this maturity in the technology as well as in the consumer. And I think Singapore is a perfect place for this. I'm very heartened to see that all of you are here because it shows an interest in this technology. The consumer is an extremely important part of this. And then there's a lot of people who are doing research, who are doing education, who are starting companies and thinking ideas. That goes hand in hand with the development and the maturation of the consumer. Okay, so that said, I think we are still in the very early stages. But I want to end with maybe just a picture of what that might be like in the future. How is it that genomics could actually integrate into your daily life? And certainly along with the theme for today of AI and I, right? If we integrate that into kind of smart city type of stuff, what would that look like? So let me start with this example. This is something that I think is pretty common. People just get sick. What happens when you get sick, though? Well, a lot of people here, you end up having to wait several hours at a polyclinic to get diagnosed, to see a doctor. Then you have to get your medication. You spend half the day there. That half the day, you're exposing other people to your coughing, to the viruses. Okay? This is not necessarily an ideal situation, maybe. When you go back to work, you have to prove that you were sick. You have to show an MC, and you have to have a doctor's note, and all this stuff, and all this has to go through HR processes. But in a world like this, where we have genomics as a daily technology, sensors and networks and AI, and a connected city, like the smart nation that we're all trying to build, what might that process look like? Well, maybe you get up and you get sick, and you pick up your cell phone, you punch a few things. A drone comes and delivers a sample collection kit straight to your house. You take that sample collection kit and you cough into it. And when you cough into it, then it goes through a process and sequences the genome, sequences the DNA that's in that sample. From there, we can tell what kind of sickness you have. From there, we can tell what kind of bacteria or virus is causing the disease. If it's a virus, then we can give you some advice. If it's a bacteria and you need antibiotics, then those will automatically be delivered by the next drone that comes in the next hour. And what you get to do, instead of going to the polyclinic, is you stay at home when you're sick and spend your time recovering and resting. That's what you should be doing. When you go back to work the next day, when you feel better, actually, let's say that you just go back to work, because when you coughed into that sample collection, we sequenced all the DNA in there. So we know it's actually you. It's validated to be you. And it all went up to the cloud anyway. So you have your DNA, you have the virus or bacterial DNA, we can show that you were sick, we know when it happened, and all that stuff can go into an automated report that goes to HR, and they don't need some paper from you to go through all this process. And that's just one simple example maybe of how things could get streamlined, and we can focus on the things that are important in a life in the future where we kind of can tie all this technology. The technology is going to be there for us to do this. And I talked about how genomics as another kind of technology that can be a daily, environmental, ubiquitous kind of monitoring system that can help us with infection, that can help us with our healthcare, that can help us with other urban applications I didn't mention, like food safety or maybe forensics as well. I really look forward to moving into this future with you guys. Thank you for showing up and showing interest in this topic, and I'll be happy to take any questions.